Hello, I'm Dr. Rhonda Souza, and this is my colleague, Dr. Stuart Speckler, and we are both gastroenterologists at the Dallas VA Medical Center and the University of Texas Southwestern Medical Center. The editors of Gut have asked us to discuss our report entitled In Esophageal Squamous Cells, Nitric Oxide Causes S-nitrosylation of Akt and Blocks SOX2 Expression. Much of this work was done by our amazing postdoc, Dr. Kiyotaka Asanuma, and Dr. Asanuma has returned to Japan to accept a faculty position, and unfortunately he can't be with us today for our discussion.
Dr. Souza and I have had a long-standing interest in gastroesophageal reflux disease, GERD, and its complication, Barrett's esophagus. Barrett's esophagus is the condition in which squamous epithelium, which is the normal lining of the esophagus, is replaced by a metaplastic intestinal-type columnar epithelium that is predisposed to develop adenocarcinoma. Barrett's is an extremely common condition. It's been estimated that approximately 6% of adults in Western countries have Barrett's esophagus, and Barrett's cancers have been increasing in frequency at an alarming rate for decades now. It's not clear how GERD causes Barrett's metaplasia to develop, but it has been shown that when you expose esophageal squamous cells to acid and bile salts, the squamous cells increase their expression of transcription factors that are involved in the development of columnar cells. CDX2 is one of those columnar cell transcription factors that increases when esophageal squamous cells are exposed to refluxed acid and bile. Now, a number of studies have focused on how GERD might cause Barrett's metaplasia by upregulating genes like CDX2 that determine a columnar cell phenotype. But it also seems likely that the pathogenesis of Barrett's metaplasia would involve the downregulation of genes involved in squamous differentiation. SOX2 is a transcription factor that promotes the development of stratified squamous epithelia, and if you genetically engineer mice so that they express low levels of SOX2, they actually develop patches of columnar epithelium in the esophagus. In humans, SOX2 is expressed in the normal esophageal squamous epithelium, but not in Barrett's metaplasia. And prior to our study, very little was known of how GERD might influence the expression of squamous differentiation genes like SOX2.
Since most studies on the pathogenesis of Barrett's metaplasia have focused on how GERD upregulates the expression of columnar cell transcription factors like CDX2, we decided to focus on how GERD might downregulate the expression of SOX2, a transcription factor involved in squamous cell differentiation. Also, studies on how GERD might cause Barrett's esophagus have focused mostly on the effects of acid and bile on esophageal cells, because acid and bile are the major toxic substances found in gastric juice. However, Dr. Ken McCall's group in Glasgow has shown that patients with GERD also can have damaging concentrations of nitric oxide in the esophagus. This nitric oxide generated from dietary nitrate found in green leafy vegetables and certain other foods. Nitric oxide reacts with oxygen to generate reactive nitrogen species, which are very toxic molecules. Very few studies have focused specifically on the role of nitric oxide in the pathogenesis of Barrett's metaplasia. So we decided to explore mechanisms whereby acid, bile salts, and nitric oxide might influence the expression of SOX2 by esophageal squamous cells. For these studies, we used two esophageal squamous cell lines, NES-B3T and NES-B10T. These are cell lines that we established from esophageal biopsies from patients with Barrett's esophagus. When we treated these squamous cells with a mixture of acidic bile salts in concentrations that can be found in reflux stomach contents in Barrett's patients, there was a significant decrease in mRNA expression of SOX2. But when we treated the cells with NOC9, which is a nitric oxide donor, there was a profound decrease in SOX2 expression, significantly greater than that produced by acidic bile salts.
Some studies suggest that SOX2 might interact with P63, another key transcription factor that is required for the development and maintenance of esophageal squamous epithelium. The p63 gene encodes two major protein isoforms called TAP63 and delta N p63. When we treated our two esophageal squamous cell lines with the nitric oxide donor NOC9, they significantly decreased their expression of both the TAP63 isoform and the delta N p63 isoform. So nitric oxide decreases expression of both of the major p63 isoforms needed to maintain squamous epithelium. Now conversely, NOC9 treatment caused an increase in the expression of CDX2, a columnar cell transcription factor. So nitric oxide decreases expression of squamous transcription factors, SOX2 and p63, and nitric oxide increases expression of the columnar cell transcription factor, CDX2.
AKT is a serine threonine kinase that can function as a key regulator of SOX2 expression. AKT is activated by phosphorylation at two of its amino acid residues, serine 473 and threonine 308. In both of our squamous cell lines, the nitric oxide donor NOC9 decreased phosphorylation of AKT at serine 473 and at threonine 308, with maximal reductions at 2 and 5 minutes after exposure. In other experiments, we also found that AKT inhibition, either with a pharmacologic agent or with RNA interference, decreased SOX2 expression, which confirms that AKT does regulate SOX2 expression in esophageal squamous cells.
So these experiments show that nitric oxide disrupts AKT signaling in esophageal squamous cells. Now, how might that happen? One likely mechanism is S-nitrosylation, the process in which nitric oxide binds covalently to cysteine residues in the AKT protein. This S-nitrosylation can alter the conformation of the AKT protein so that it can no longer function as a kinase. To confirm that nitric oxide causes S-nitrosylation of AKT protein, we performed a Biotin-Switch assay. In the Biotin-Switch assay, we took esophageal squamous cells that were exposed to NOC9 for up to 5 minutes, and we treated them with ascorbic acid. This ascorbate treatment converts S-nitrosylated cysteine residues to thiols that can be labeled with biotin. So you switch biotin for S-nitrosothiols, hence the name Biotin-Switch. Notice that when we treat these cells that were exposed to NOC9 with ascorbic acid, we get a strong signal for biotinylated AKT. This confirms that nitric oxide causes S-nitrosylation of AKT protein.
This slide summarizes our findings on how nitric oxide generated from dietary nitrate might contribute to the pathogenesis of Barrett's metaplasia. Nitric oxide in the esophageal lumen causes S-nitrosylation of AKT, which blocks its phosphorylation at threonine 308 and serine 473. This S-nitrosylated AKT can no longer activate the SOX2 gene, leading to decreased expression of SOX2 mRNA and protein, which inhibits squamous differentiation. NO also decreases the expression of the TA and delta N isoforms of p63, which also inhibits squamous differentiation. At the same time, nitric oxide increases the expression of CDX2, which promotes intestinal differentiation. Conceivably, this combination of events might result in the development of Barrett's intestinal metaplasia.
We have demonstrated that nitric oxide triggers a series of molecular events in esophageal squamous cells that could inhibit squamous differentiation while simultaneously promoting intestinal differentiation. This combination of events might contribute to the pathogenesis of Barrett's metaplasia. So our study provides a rationale for research on interventions that might reduce nitric oxide production in the distal esophagus as a way to prevent the development of Barrett's esophagus and its most dreaded complication, esophageal adenocarcinoma. Dr. Souza and I thank you for listening to us today. Goodbye.
Goodbye.